Great, we will wait for a couple of minutes to let others join. if we can start with just share. So good evening, everyone, and welcome back to session 10 of our online course, Strategy Physical Engineering. And we have been discussing about the last year's assignment in order to prepare you guys for the exam that will be coming up at the end of the course. So let's look at some of the topics that have been covered in week 10. Have any of you gone through the week 10 lecture? Am I audible? If one of you could respond, just wanted to know if I'm audible. Uh, can all of you hear me? No? Okay, assuming you guys can hear me. If not, then anyone of you can just unmute your mic and let me know if you're audible. Okay, so in previous 2 weeks, we have been looking about adaptive immunity, which comes into play when your innate immunity is not able to get rid of the incoming the foreign material or microorganisms. And as we have discussed in the last session, that your antigen presenting cells, which is nothing but your dendritic cells and macrophages, are linked in between your innate and adaptive immunity. And so these are they phagocytose the foreign material or microorganisms, and they present antigens on the surface, which are then identified by your B cells and T cells, which are part of the adaptive immunity. And T cells, as you all know, have their major function is to generate antibodies against the foreign material. And there are several classes of T cells, such as cytotoxic, helper, and regulatory T cells, the details of which have been covered in the lectures in week 10. Going ahead, then we have discussed about vaccines. Vaccines, as you all might be knowing, is nothing but a pathogen or a part of pathogen that is delivered to generate memory immune response in order to strengthen the actual immune response in case of an infection. So vaccines are nothing but you deliver a part of pathogen uh, to any route, and then that is recognized by your body as a foreign material, and then your body mounts an immune response against this pathogen or a part of pathogen, and then so a lot of immunology happens in the body, we won't be going into detail. You can refer to the lecture. So whenever there is an infection in the body, your body is going to generate antibodies against it. Most probably your B cells will generate antibodies, and they will try getting rid of this infection. Now, after they have done their job, so most of the B cells will be cleared out after the infection has been cleared out from the body. But a part of these B cells will remain in the body as memory cells, and then they can remain in your body for a period of 5 to 10 years. And then whenever you get an actual infection, so vaccine is nothing but it's a part of pathogen, which is not an actual infection. So in case when you get an actual infection from the same virus or the same bacteria, then your memory cells can come into play, and then they can get rid of that infection in quite a shorter amount of time than it would have taken in case you did not have the vaccine. So in this way, your body is protected from the infection, and that's why vaccines are used to stop epidemic infections such as smallpox, etc. You guys might be knowing more about vaccines in case of COVID. So there are different types of vaccines depending on when they are given. So one type is prophylactic vaccine, which is given before the disease develops. Another type is therapeutic vaccine. In this case, the patient is already suffering from a disease, and then you give a vaccine in order to strengthen the immune response. Sometimes the patient's body is not able to have a strong immune response against the pathogen. So that's why administered therapeutic vaccines in that case. Next type is live attenuated vaccines. Now, these four types are based on what actually are you administering as a vaccine, what part of the pathogen are you giving. So in case of live attenuated vaccine, this is a living organism that you are administering. For example, let's just say there's a smallpox vaccine you want to develop. In that case, you take the smallpox pathogen, you will attenuate it. Attenuate in the sense it's a living organism, but then you have removed its infection, remove the part of the pathogen which actually causes the infection, and then that's what you administer. So in case of live attenuated vaccine, you get a very strong immune response from the body, but there's also risk involved in these type of vaccines because you even though you have removed the virulence factors, this is still a live organism, and once you store it or once you administer it, they can regain their virulence and they can actually cause the disease which you're trying to prevent in the patient's body. So that's about live attenuated vaccine, and then you have something known as inactivated vaccine. In this case, the organism is killed, you have inactivated the organism, and then you have administered it as a vaccine. In this case, although the organism is not alive, it will still have its protein, it will still have its DNA material there, and that's what the immunity is generated against. Another type is subunit or recombinant vaccine, in which you take a part of, instead of giving the whole pathogen as a vaccine, you take a particular part, a subunit of this pathogen. For example, it might be a protein which is flagged on the surface. So that can be administered as a vaccine. So in case you have an actual infection from the same pathogen, the same protein can be recognized by your immune system and by the memory cells, and then they can mount an immune response. Another type is toxoid vaccines. In this case, you identify a toxin. For example, in several pathogens, the actual pathogen might not cause infection, but a certain particular toxin which is released by this bacteria can be toxic, can cause infection in the body. So what you can do is instead of giving the entire organism as a vaccine, you can identify this toxin and then you can administer this toxin as a vaccine. So whenever there is an infection by this pathogen, the pathogen will start releasing this toxin and that will be recognized by your body, and then the immune response will be against this toxin. So these are a few types of vaccine, and then going ahead, particle-based delivery for vaccines was discussed in this week. So now you have to look for a method to deliver this vaccine. Let's say you have designed a very good vaccine, and now you're looking for delivery of this vaccine into the patient's body, into the patient's body. Yeah. So there are several particle-based delivery methods for vaccine. So now why do we want to go for particle-based? Why not directly deliver the vaccine? It's because particle-based delivery has certain advantages. First is it has higher uptake by ATP. So let's look at this concept in a big detail. So whenever you are giving a vaccine, let's say you have identified a protein, and that protein is what you want to administer as a vaccine in a patient's body. Now that protein is going to be of a very small size, let's say somewhere around in 5 to 6 nanometers. If you administer this protein as it is, if you inject it, there are high chances that this will get cleared out from the body very easily because it's a very small protein. And your antigen presenting cells, which are macrophages and dendritic cells, they have a certain size range of what particles they can uptake. Now, if the protein is very, very small, they won't be able to uptake this particle very easily, and most of it will be cleared out, so that won't be a very good vaccine delivery. So what you can do, you can take this vaccine, you can put it in a micro or a nanoparticle, let's say of somewhere around size 200 to 300 nanometer, and then you can administer it into the patient's body. Now, this being a larger particle, your ABCs, your macrophages and dendritic cells will clearly identify this as a foreign particle, and then they will start uptake. So you're increasing the uptake of this vaccine delivery. Also, another advantage of delivering vaccines in particles is co-delivery of adjuvants. So adjuvants are nothing but molecules which help in increasing the immune response, they enhance the immune response. These are required for co-stimulation of the antigen presenting cells. So along with your actual vaccine subunit, you can also put in adjuvants in the same particle and then deliver it to ATP. So this will help in co-stimulation. And also another advantage of particle-based delivery for vaccines is they can carry all types of antigens. For example, if you have designed a DNA or an RNA as a vaccine, they can easily be incorporated into a particle and then you can deliver that rather than delivering free DNA or free RNA. So These are some of the advantages of particle-based delivery for vaccine. Another important concept that was discussed was immunoisolate cell therapy. So this is nothing but instead of now delivering a drug, so we have been looking at drug delivery for quite a lot of lectures now. So instead of delivering a drug, you are actually delivering cells. So in this case, your cells are going to be the drug. So what you can do is, so this is nothing but transplanting cells which are producing a therapeutic protein. For example, let's say you have encountered a patient and the patient lacks a very important protein. His body is not able to make that protein. So what you can do is you can take cells from a different donor which actually produce that protein, and you can put the cells in a membrane. You can put it in a polymer membrane, and then you can put this entire assembly into the patient's body. So this is known as immunoisolated cell therapy. Now you can design the polymer membrane in such a way that it will have a very small pore size. It will just allow the therapeutic protein to release from this encapsulation, but it will not allow the larger cells, that is the immune cells of the patient's body or the antibodies to come in. So now you have isolated this entire cell mass from the patient's immune system. So that is the advantage of immuno-isolated cell therapy. It will not lead to immune rejection. And then the last concept that was discussed in this week was gene therapy. Gene therapy is genetically, so gene therapy is nothing but you can change the genetic makeup of few cells in order to relieve the patient of a disease. In this case, you can actually inject genetically modified cells to produce few proteins which the patient might be lacking. Or in some cases, let's say the patient is not having the gene that is required to produce that protein. In that case, you can add a new gene to the patient's body, or you can suppose there's a faulty gene. So the protein is being produced, but it is faulty in nature, because of which it is not able to carry out its activity. So rather than giving the protein, you can actually replace that defective gene by the correct sequence of gene, or else you can repair this gene. So these are few of the concepts that were discussed in this week. So having this background, we can now go ahead with the assignment and see if it is solved. You guys have any doubts? You people are able to hear me, right? Can anybody just confirm if I'm audible? I hope you guys can hear me, because I don't know, none of you are responding. So I'm just going to go ahead with the assignment questions now. So the first question is, in immunoisolated therapy, the cells should be within some distance from the nearest blood vessel to facilitate effective diffusion of nutrients. So can any of you let me know the answer? Okay, no one. So this is the answer. The correct answer is within 500 microns. So as I said, in immunoisolated therapy, you have your cells in a polymer encapsulated membrane, and you are injecting this into the patient's body. Now, as these are living cells, they will definitely be metabolizing, and then as a result, they'll require nutrients. So to facilitate effective diffusion of nutrients, whenever you inject this into the patient's body, it should be within 500 microns of the nearest blood vessel. Because if it's far away from the blood vessel, then we will have diffusion limitation. The oxygen and the nutrients won't be able to effectively diffuse. As a result, the cells will start dying, and then it will lead to therapy failure. So that's why in immunoisolated therapy, the cells should be within 500 microns from the nearest blood vessel. Is this clear? Does anybody have a doubt? not, then we'll just go ahead with the next question. Okay, the next question is which of the following would antagonize immunoisolated therapy, and you have to choose two correct options. I'll wait for you guys to read through the options, and if anybody wants to volunteer, just unmute your mic and let me know. Okay, Noel. So antagonizing meaning the question wants to the question means that which of these four lines will lead to a failure in immunoisolated therapy, meaning which of this is not suitable for immunoisolated therapy. So the correct answer is use of biodegradable polymer to encapsulate the cells. Let's just look at the options one by one. So the first is cells that were grafted within 200 to 500 micrometers from the vasculature. As we already saw that this is the correct distance from the vasculature in which you can actually implant your immunoisolated therapy cells. Next option is use of biodegradable polymer to encapsulate the cells. Now, the whole purpose of immunoisolated cell therapy is to isolate the cells from the patient's immune system. So, because of since we want this, the polymer which you are going to use for immunoisolated cell therapy has to be non-erodible. If it's biodegradable and if it starts degrading, then it will lead to a larger pore size, and then this will cause all your immune cells from the patient's body to enter into the encapsulation, and they might start creating immune response against the cells which we have implanted. So that's why the membrane which is encapsulating the cells should be of a non-erodible polymer. So as a result, first, the second option is something which will antagonize the immunoisolated therapy. Third is use of mechanically stable non-erodible polymer to encapsulate cells, like we discussed right now. And fourth is use of a biocompatible polymer that does not allow fibrosis of the implant to encapsulate cells. So this is what we actually want. If your polymer is not biocompatible, the patient's body will start fibrosing over this encapsulated cells. As a result, it will create diffusion problems and your cells will not be able to function properly. Next option is use of a polymer that can filter out antibodies and T cells to encapsulate cells. That's actually what we want. That is the whole purpose of immunoisolated therapy. Next is use of allogenic or autologous cells for encapsulating an implant. So the cells which are used in this encapsulation can be taken from the patient's body or can be taken from a donor. Next is use of a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells. This is what we don't want, because if we use a polymer that is allowing free exchange of proteins and cells, then the patient's immune cells and the cytokines will enter into this encapsulation and they can lead to an immune response. Last option is use of a polymer that allows free passage of small proteins but not cells in large complexes to encapsulate therapeutic cells. So this is again what we want. We want the polymer to be designed in such a way that it can allow free passage of small proteins. So these small proteins are nothing but the nutrients which will be required by the cells to function, and your therapeutic protein which we need the protein to come out of that encapsulation and into the patient's body. So the right answer for this question is use of biodegradable polymer to encapsulate the cells and use of a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells will actually antagonize the immunoisolated therapy. Is this clear? Does anybody have doubt in this question? not, then I'll move on to the next question. Okay, so the next question is the process of weakening a pathogen by generating a vaccine is called, and these are your options. If anybody wants to volunteer, you can let me know. If not, I'll give you guys some time to reflect over this.
I I think my Bluetooth was on and I was not able to hear you. Can anybody repeat if somebody has answered for this question? Is anybody trying to answer?
Second option, attenuation.
Okay, yeah. Thank you. So I'm sorry, my Bluetooth was on for the laptop. I couldn't hear you people if you were answering previously. If you guys have any doubts which I might not have heard, you can ask me again. So yeah, you were correct. The right answer is attenuation. So pathogen attenuation is a process where we decrease the efficiency or virulence of the pathogen by either heating it or by treating it with some chemical reagents. So this is done in live attenuated vaccines where we want to reduce the virulence of the organism and then administer it as a vaccine. Is this clear? If yes, then I'll move on towards the next question. The next question is, what are the major challenges of immune isolated cell therapy? And these are your options. You guys can let me know when you are ready to answer. No one? Okay, then I'll discuss the answer. So the right answer for this question is generation of immune reaction, fibrosis of implant over an extended period. So these are the major challenges of immune isolated cell therapy. Even if you have the cells which are used in this isolated cell therapy might have been taken from the same patient, still there are chances that an immune reaction can be generated against these cells. And the other problem is fibrosis of implant over an extended period. Now since this is a foreign material for the patient's body, where you have your cells in a polymer membrane and you have implanted this at some location in the patient's body. So over an extended period, the patient's body is going to consider this as a foreign material. It will start attacking this isolated cell therapy. Since it's a very big molecule for the immune system to clear away, it will start fibrosing over it. It will just create fibrosis on the implant surface, and it will just isolate it from the patient's body. So and then this can lead to a diffusion problem. So these are two major challenges of immune isolated cell therapy. Is this clear?
Yes, ma'am.
Yeah, thank you. Then I'll move towards the next question. The next question is, which of the following methods can be used for encapsulating cells in biomaterial? We have to choose two correct options. Any volunteers? Okay, no one? Okay, let's discuss the answer. The right answer for this question is ionic gelation and microfluidics. So these are two methods which can be used to encapsulate cells in biomaterials. Ionic gelation is nothing but it makes use of alginate and calcium solution. So let's say you have a device like this, in which you have your alginate suspension, and this is where you have your cells in this sodium alginate suspension, and you have loaded this in a syringe, and then you have a calcium chloride solution. So you can start putting these cells in this alginate suspension slowly, drop by drop, into this calcium chloride solution, and the calcium and the alginate will then form a network, and this will polymerize the solution. And since as it is getting polymerized, since you have free cells suspended in this solution, they will be entrapped into this polymer solution, and that's how you can have the encapsulated cells which can then be implanted. So this is one method. This is known as ionic gelation. Microfluidics is nothing but you make use of microfluidic devices to again do the same thing. You can entrap the cells inside these polymer membranes. The other methods, which is spray drying or emulsion based methods or hot melt, are actually not used for encapsulating cells, since these require a lot of energy. For example, in hot melt, you have higher temperature involved. This can damage the cells, which is what you don't want. Also, in emulsion based methods, if you have emulsions, which is nothing but oil in water emulsion, or water in oil and again in water, so this requires some kind of solvents. So these solvents might be harmful for the cells. They can start denaturing your cells. So these methods are not used for encapsulating cells in biomaterial. The only two methods are ionic gelation and microfluidics. Is this clear? Okay, then let's move on towards the next question. The next question is, which of the following is a type of therapeutic vaccine? And we have to choose two correct options, and these are your options. So if anybody knows the answer, let me know.
Polio, smallpox vaccine.
Polio and smallpox vaccine. Okay, anybody else? Thanks for answering.
Cancer vaccine and rabies vaccine.
Can you please repeat? One answer is rabies vaccine. Okay, and
Cancer
okay.
vaccine.
Yeah, I got it. Thanks for answering. Anyone else? Okay, no more responses. Then let's look at the answer. So the right answer is cancer vaccine and rabies vaccine. So the one who answered the first, which was polio and smallpox vaccine, both of these would have been correct if it was a prophylactic vaccine. So both polio and smallpox vaccines are given even before the human develops the disease. So this is mostly administered to babies so that they don't develop polio and smallpox disease later on. So therapeutic vaccine, as I have discussed previously, is nothing but it is given after the disease, and the purpose is to strengthen the immune system to fight back against the active disease. So cancer vaccines are prominent example of this. Rabies vaccine can be considered as a therapeutic vaccine, but technically speaking it is also a prophylactic vaccine. So rabies vaccine is given when the patient has already has rabies, but we want to prevent the pathogen from entering the brain of the patient, so that's when rabies vaccine is administered. So it can be considered as a therapeutic vaccine, but technically speaking it is still a post exposure prophylaxis vaccine. So for this for the purpose of this question, both the answers are correct: cancer vaccine and rabies vaccine. Is this clear? Okay, then let's move on towards the next question. Next is, which branch of immunity is triggered when a vaccine is administered to an individual? Two correct options. Any guesses?
Humoral and adaptive.
Okay, thanks for answering. Anybody else?
Adaptive is okay, and another one.
Okay, one option you have chosen is adaptive. Okay, thank you. Okay, the correct answer is humoral and immunity. So the one who answered first, you were correct. So both of these are so humoral immunity is nothing but the B cell response which gets generated, the antibodies which are generated by B cells. So whenever a vaccine is administered, both these arms of immunities get activated, the humoral, because antibodies will be generated against that vaccine, and adaptive immunity, which consists of T cells, wherein your APCs will phagocytose this vaccine and they'll present it on their surface because they're antigen presenting cells, and this antigen will then be recognized by the T cell receptors, and then that will lead to a cytotoxic T response. So that is nothing but the adaptive immunity. So whenever you administer a vaccine, both humoral and adaptive immunity will be activated. Okay. Let's look at the next question then. The next is, what is an adjuvant in the context of immune response? And these are your options. So if you guys have read through the material, you might be able to answer this. Any volunteers?
A and the last one.
No, you have to choose only one. So this question will have only one.
The last one.
Okay, that does not affect the immune response whatsoever. Okay, that's one response. Anybody else? Sorry?
Second option, a molecule that stimulates an immune response
To
to
target
target antigen.
Okay, that's a response. Anybody else? Thanks for answering. Okay, no one. So yeah, the correct answer is option 2. An adjuvant is a molecule that stimulates the immune response to target antigen. So adjuvants are nothing but molecules which are administered along with the target antigen so that it strengthens the immune response, which is nothing but it amplifies the response, and it can be used to direct the response towards a certain type, either cellular or humoral. And this apart from doing this, it has several other functions as well. So whenever you are administering a vaccine, you have to make sure that the immune response generated against that vaccine is strong and that it stimulates the antigen presenting cells also. In case the antigen presenting cells are not stimulated and only antigen is present, then that will actually lead to immune tolerance, which is what we don't want. Immune tolerance is nothing but, let's say you have a foreign antigen, a bacterial protein, and you have given that as a vaccine, and then your APCs have phagocytosed it and presented it on their surface. Now if the APCs are not stimulated, your T cells or your B cells are going to consider this antigen as a self antigen, meaning it will consider that your body has generated this antigen, and as a result it will not show any immune response. Now in case of an active disease, if this bacterial infection happens, then your body, since it does not have any memory, it does not consider this as a foreign material, it is going to consider that bacteria as a self antigen. So this will actually lead to immune tolerance, and the bacterial infection will then proliferate in the patient's body. So that's why you need an adjuvant which will strengthen the immune response and stimulate your antigen presenting cells. Making sense?
Yes, ma'am.
Yeah. Thank you. If there are no doubts in this question, we'll move on towards the next question. So the next is, which of the following are undesirable properties of polymer used for an immunoisolated implant? So we discussed some of these concepts in the previous questions as well. So 2 correct options here. Any guesses?
I think so. Toxic and biodegradable.
Okay, that's one response. Thanks for answering. Anybody else wants to give it a try? Okay. Yeah, you were correct. The right answer is toxic and biodegradable. We don't want the polymer membrane to be toxic, of course, because we are injecting this into a human body and we don't want it to be toxic. Next is, we don't want it to be biodegradable because, as we discussed, if it starts degrading, it will allow the immune cells of the patient's body to enter into this encapsulation, and then that is not a good thing. It will lead to the implant failure, and we have to remove the implant in that case. So the undesirable properties of a polymer is, it should not be toxic, it should not be biodegradable. The last question of this assignment is, which of the following is a potential disadvantage of particle based approaches while delivering vaccine to antigen presenting cells? And we have to select 2 correct options. So I'll give you guys some time to read through the options. Okay, no one? Then let's look at the answer. The correct answer is last 2 options. The formulation can be unstable, or the antigen might lose integrity in the process of encapsulation. Since this is a vaccine, the vaccine could be anything. It can be a protein or it can be a DNA. And whenever you're, let's say you have put this into a particle and then you are delivering it so that it is engulfed by the antigen presenting cells. Now let's suppose it's a DNA, then we are not sure. It can be an unstable formulation also, because DNA being, there are a lot of things which can denature the DNA. So as a result, the formulation might be unstable, and also the antigen might lose integrity in the process of encapsulation. Let's say it's a very sensitive protein, and in the process of encapsulating it, we have caused its shape or conformation to change. In that case, it might lose its integrity. It is no longer the antigen which the bacteria might actually have, and as a result, the immune response won't be generated against the actual antigen. So these are some of the disadvantages whenever we are trying to deliver vaccine in particles. So that's the last question of the assignment. If you guys want me to discuss any other question, people who have joined late, if you want me to go through any of the questions, repeat some of this, we can do that. Anybody wants me to repeat?
Can you repeat 1 to 5?
Yeah, sure. Let me just go over it once again. So yeah, the first question is, in immune isolated therapy, the cells should be within what distance from the nearest blood vessel to facilitate effective diffusion of nutrients? So the correct answer was the cells should be within 500 micron, because in immune isolated therapy we have this polymer membrane, we have our cells inside that, and this entire implant we are injecting into the patient's body. Now if it is very far from the blood vessels, we can there will be diffusion limitations, because these are living cells. They need oxygen and they need nutrients as well. So if they are very far from the blood vessel, we will actually cause diffusion problem. So as a reason, they should be within 200 to 500 micron from the nearest blood vessel. So that's the design requirement for immunoisolated cell therapy. Next question was, which of the following would antagonize immunoisolated therapy, meaning which will be a disadvantage to this therapy? And the right answer was use of biodegradable polymer to encapsulate the cells, which is what we don't want, because if the membrane starts degrading, the polymer starts degrading, it will allow the immune cells of the patient's body to attack the cells which are encapsulated in this implant. And the next was use of a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells. Again, the same problem. If the membrane is porous enough to allow proteins and cells to enter, it might let the immune cells of the patient's body to enter the encapsulation, or certain cytokines can enter it, which will then trigger the immune response, which is what we don't want. So these two options were correct for the question. Next was, the process of weakening a pathogen while generating a vaccine is called as attenuation. This process is used in live attenuated vaccines, where we are trying to decrease the virulence of the pathogen and then give this pathogen as a vaccine in the patient's body. So it will not actually lead to a disease, but your immune system will remember this pathogen, and then in case of an active disease, when the actual infection happens, it can mount a stronger immune response. So attenuation is nothing but decreasing the efficiency or the virulence of the pathogen. Then next question was, what are the major challenges of immune isolated cell therapy? First was generation of immune reaction. Even if the cells used in immune isolated cell therapy are from the patient's own body, there are chances that it might lead to generation of immune reaction. And fibrosis is another problem, because the patient's body is not able to clear off this implant, which is still a foreign entity for the body. So as a result, it will start fibrosing over this implant, and that will cause diffusion problems. So that will lead to therapy failure. So these are two major challenges for immune isolated cell therapy. And then fifth question was, which of the following methods can be used for encapsulating cells in biomaterials? The two methods widely used are ionic gelation and microfluidics. Is this clear?
Yes, ma'am. Thank you.
Yeah, welcome. I guess you people have no more doubts in that case. Yeah, anybody trying to say anything? If not, I'll stop the meeting and we guys can meet next week. Dhanashree, you have a problem? Yeah, sure. Thank you.